last week we have come to study about the coccus. So coccus is a big group of the bacteria, which it includes lots of the pathogenic bacteria which do harm to our human being. So when we learn about the microbiology, the bacteria, it is the gram staining is a very important method to distinguish the bacteria. So I think after we learn about the cocci, we'd better to learn how to do the gram staining. So here, I have a PPT for our lab class, and this PPT is about the lab 2, the second experiment, and it concludes 2 parts. The first part is about the microbiological culture medium. So it is how to prepare medium for culture the bacteria and how to do the sterilization about the medium. However, this part, I think it is for self-study. I will send the PPT to our DINTalk group, and then you can you can read the slides. And what's more, I will send some link to show you how to make the medium and then how to do the sterilization, as we just have online class for experiments. So we have limited method to guide you to teach you to do the sterilization, because sterilization just need to use some machine. You just need to put the medium inside the machine and you turn on some program and and wait it for 1 hour or maybe 1 and 2 hours, and after it cool down and you take it out, then it's done. So just like this. So we will skip the part 1, and today we will learn about the part 2, the gram staining. Gram staining. This is really important. So here in the slides, there are learning objectives, just like every experiment. I also sent you the document for experiments class, and there are learning objective. That is to understand and the mechanism and understand how to do the gram staining, okay, and perform satisfactory gram stain. And there are also materials, and the principles for this experiment. So you can write it down on your lab report about these 3 slides. And here, what's more, I want to show you more important, that is beside our slides. I want to show you something here. Sorry. Oh. This one. It's a general introduction for gram stain. And what's more, in this website, it shows us what is a gram stain and how to collect the sample and so many things. So I think it's better we can learn it, just on the website. It's better. So first, what is a gram stain? The gram stain is the most common used laboratory test just to check the bacteria. Okay, check the bacteria. So all kinds of bacteria, the first test, the most easy test for them, it is the gram stain. and the bacteria came from the suspected infection or in some certain body fluid. And it can give a quick result. Then the doctor, they can know if the bacteria are present. So it means, bacteria is really small, and under the microscope, and maybe some student says we can observe it under the microscope. However, if without staining, the bacteria doesn't has any color. It is transparent. So it's unbelievable that you can see the bacteria just under the light microscope. Only when it is been stained with some color, then you can notice it. You can see the shape, the morphology of the bacteria, and the structure of the bacteria. So the gram stain is a really quick and simple test to know if bacteria are present, and even the general types. It means which kind of bacteria it may belong. Does it belong to coccus? Does it belong to the rods, the bacillus, or just the spiral? Okay. So this will help guide further identification test and treatment options. Because in the lesson of the cocci, we learned this last week. We know that some of the cocci, they are resistant to some kind of antibiotics, right? So if you know the gram-positive bacteria or the gram-negative bacteria, then they can help you to guide the further identification test, and also to help you to judge which kind of treatment is much better for the patient. Okay. And here it introduces the bacteria that we have learned it, right? So some types bacteria are harmful less. It means they are not harmful or even beneficial or even do some good to human beings. But the other bacteria can cause infections and disease. So the gram stain, it helps diagnose the harmful bacteria. Okay. And what color of the gram stain it will show to us. So under a gram stain, different kinds of bacteria will change one of two sets of colors. It means some of them are pink to red, or some of them are purple to blue. So pink to red, they belong to a group. And purple and blue, they belong to the other group. Yeah. So under the special series of stain, they can show the color like pink or red or purple or blue, and they can be grouped as gram-positive or gram-negative. However, the gram-positive or gram-negative doesn't mean it is good or bad. It just shows, as we have learned, the cell wall of the bacteria. It just shows the different cell wall has different reaction to the reagents. Okay, so this is the general introduction for the gram stain. And what's more, here, not all of the bacteria can be tested using the gram stain method. And the gram stains doesn't usually provide a diagnosis alone. So it means some of the bacteria, they need much more tests, or we need to combine different kind of diagnosis together, and then finally we will get a result to determine the features, the characteristics of a certain bacteria. Okay, and then gram staining is very essential. Essential means important. It's really essential, important, and popular staining technique. Yeah, staining technique in microbiology that when it was found in, you see, in 1882, yeah, this scientist, yeah, he investigates the method, the technique for gram staining, and it was found in 1882. And nowadays, people still use this method so often. Yeah, in the lab, both in lab and in the hospital. And the second part is introduce what are gram-positive and what are gram-negative bacteria. I think we have learned about it, right? The gram-positive and gram-negative bacteria. However, here I still find a video, yeah, a cartoon video. Sorry. I think it's a advertisement. Yeah. Sorry, not this one. It should be this. Yeah, this video, it will help.
The gram stain is named after.
Oh, where is it? Sorry. Let me just find it again. I try to find a video which will help you to think about the gram positive and gram negative bacteria.
Bacteria have cell walls made up of polysaccharides that give them strength and rigidity. This is important since bacteria often experience variations in osmotic pressure due to the different solutions they encounter, and it is their cell walls which prevent them from shrinking or swelling. As a reminder, osmosis is the process by which solvent molecules pass through a semi-permeable membrane from a less to the membrane. Peptide polysaccharide layer. All cell walls have a peptide polysaccharide.
So here, yeah, this shows the bacterial cell wall, right? And it has a peptide glycogen. Yeah. And this is the structure
layer called peptidoglycan, also known as murine. Peptidoglycan is a polymer made up of sugars and amino acids which forms a kind of mesh. Bacteria can be classified based on their reaction to the gram stain, which identifies them as gram-positive or gram-negative, based on the chemical and physical properties of their cell walls. Gram-positive bacteria have a thick cell wall, which consists of up to around 30 layers of peptidoglycan. This cell wall surrounds a monoderm, which is a single plasma membrane. Gram-negative bacteria have a much thinner cell wall, consisting of a single layer of peptidoglycan. This layer of peptidoglycan is sandwiched between two lipid bilayer membranes called diderms. We can differentiate between gram-positive and gram-negative bacteria by dyeing them with crystal violet.
Okay, so just now it shows the gram-positive and the gram-negative, the structure for them, right? So we have learned about it. So I just show you a short video about the gram-positive and gram-negative. And then here, followed by the gram-positive and gram-negative bacteria, the definition for them. Here, it just displayed some examples for gram-positive bacteria. Yeah, and here we see, yeah, we just learn about the Staphylococcus species. Yeah, and we also learn about the Streptococcus coccus. So on the first day, I think when I introduced the structure, the shape of the bacteria, I told you the difference between the staphylococcus and the staphylococcus and the staphylococcus. So, one of them are just form something like a grape. It's a cluster of the bacteria, and another one is just a form like the beads. Yeah, just like a necklace to show, look like a chain, right? And I also give you examples for the Listeria bacteria, because Listeria bacteria, it causes contamination to food, especially even food stored in the refrigerator. Yeah, it can still be polluted by this Listeria bacteria and cause the diarrhea for the patient. So here it just shows some typical gram-positive bacteria. Yeah, so it includes something like this. And then here are the gram-negative bacteria, such as E. coli. Yeah, E. coli belong to the not the coccus, but it belong to the rod, the bacillus. Yeah, bacillus. And so after that, it shows the difference between a gram stain and a bacteria culture. So this part, I think you can read by yourself. And then here I just want to show this one. What conditions do gram stains help diagnose? So when you go to the hospital, if you have some infection, and the doctor want to decide if this infection is caused by bacteria or caused by viruses. So maybe they will do a simple gram stain, and to collect the sample, so they need to get the samples. So later on, I will introduce which kind or which area does the doctor can get the sample to do the gram staining. However, here it just show, you see here, it just show common gram-negative bacteria and their associated conditions. Actually, their associated conditions means the disease caused by them. So it just give these examples. You see, so as we learned just now, the gram-negative bacteria include the E. coli, right? And E. coli can cause the urinary tract infection. So this caused by E. coli. And it can also cause food poisoning. Food poisoning means, when the food is polluted or contaminated by the E. coli, and you eat this kind of food, then you will feel toxic to your digestive. You will get digestive infection. It will cause vomiting and diarrhea and even some fever, something like this. And this one, the Legionella pneumonia, it will cause, the most common symptom for this bacteria is just pneumonia, and some other related symptoms. And here it also contains the Neisseria group. Okay, so this is the gram-negative bacterial samples. The examples for the gram-negative bacteria and the disease caused by this kind of bacteria. I also put the link. I write down the link, in our slides, And so I will send the slides to you, and then if you want to have a review or you want to start it again by yourself, then you can just open, click the not click, but you can paste the link to your website and open it and read it. So here, here are the gram-negative bacteria, and these are the gram-positive bacteria, and their associate conditions, associate disease caused by, such as the streptococcus pneumonia, and it will cause the bacterial pneumonia. And the staphylococcus, and also it's this kind of coccus, it can also cause the bacterial pneumonia, and what's more, it causes many other associated symptoms, and even the toxic shock syndrome. So this bacteria sometimes it is fatal, and the Listeria bacteria, as we introduced just now, it's also food poisoning. Okay, and then after we gain a little more impression about the gram stain, so we know why do we need to do the gram staining, and what is the importance of the gram staining, and how does the gram staining help us to distinguish? So the gram staining just help us to distinguish the gram-negative and the gram-positive bacteria. And these two groups of bacteria, we will remember the most common species which belong to different group and the disease caused by them. And after that, the most important is when do we need, and what's more, let me see the samples. How do I prepare for a gram stain test? So when is a gram stain test performed? So we can say that the healthcare providers typically request a gram stain along with a bacterial culture when they suspect you have a bacteria or sometimes a fungi infection. So it's one of the most common ways to quickly diagnose bacterial infection in the body. So that is when the doctors suspect that you may get a bacterial infection, then it's better they collect some samples and then to do the gram test. And here, to collect the samples, so the steps to perform the gram stain test, there are 3 general steps. The first is collect the sample and processing the sample and examine the sample. So first, let's see how to collect the sample for the gram staining. And then after that, then we will see the video. We will see the video just to show you how to do the gram stain. Like this. So first one is collect the sample. A gram stain test will need to collect a sample from the site of suspected infection. So there are some ways we can collect the sample. First is brushing or scratching tissue from the surface of a part of your body. So just like if your finger get cut by the knife, or if you have some wound on your arms or your leg, which get infection, so they just need to use a cotton stick to brushing or scratching the tissue from the surface of your body. And the second option is collecting the fluid or discharge sample from your body. Yeah. The third one is using a needle aspiration to draw the fluid sample from an area in your body. So these are the three common ways to collect the samples. And the samples can get from the throat, nostrils. Yeah. If they want to get sample from nostrils, so they just scratching some tissue in the nostrils, right? And sometimes they can collect samples from the urogenital tract, and then or as well as the skin wound. Skin wound means to get the tissue from the surface of the body, right? So the samples can also be collected by the sputum, the spit, or even the urine and the stool. Yes. And because the bacteria, they can grow almost everywhere. So the sputum, the urine, the stool, it also can contains the bacteria. And some other areas in your body that may require the use of the fine needles. Fine needle aspiration, it includes the synovial fluid in the joints, and the fluid around your heart, fluid around your lungs, even the fluid around your spinal cord. That is the cerebrospinal fluid. So you see, it just gives you examples of three different sites which you can get samples, three different ways where you can get samples. So when we get the sample, then how to do the Gram staining? Then just here it will show this video. It show the Gram stain method very well. Now let's watch the video.
The Gram stain is named after its inventor, Hans Christian Gram. As a Danish botanist and teacher of pharmacology, he devised this important method of differentiating bacteria in 1884 that is still in common usage today. The Gram stain is used to distinguish between gram-positive and gram-negative cells. It is the most commonly used.
Okay, so just now we have learned that the Gram staining will make the bacteria different color, right? Some of them are pink to red. Some of them are blue to purple. So gram-positive, they are blue and purple. And gram-negative, they are pink and red color. This is the most common use method
to observe bacteria. The Gram stain provides two important features: the Gram reaction, or color of the cell, and the cell morphology, the shape of the cell. Bacterial cells can be spherical, rod, or corkscrew in shape, and nearly all clinically significant bacteria can be differentiated into these three groups. Knowing which type is causing an infection is critical to patient care. The Gram stain differentiates cells based upon differences in the structure of the cell wall. The cell wall is made up of peptidoglycan, which consists of sugars and amino acids that form a layer around the plasma membrane of most bacteria. Bacteria that stain dark blue-purple are called gram-positive. They have a thick peptidoglycan layer that retains the primary dye. Conversely, bacteria that stain pink to red are called gram-negative. They have a thin peptidoglycan layer that loses the primary dye when flooded with alcohol. The Gram stain begins by adding 1 drop of sterile saline or deionized water onto the center of the.
So So here, it shows the first step. Before you put the sample into the slide, first you need to take a glass slide. It should be a dry glass slide, and put some saline or some sterilization water. So saline or saline is most recommended. Yeah, put a drop of saline on the center of the
slide. Then take a sterile loop and touch the edge of an isolated colony.
Okay, then you see this person. Yeah, sorry. You see here is a plate, a culture plate for bacteria, and he opened.
Then take a sterile loop.
Then he opened the plate, and you see he used a tool. This tool has a circle on the on the top, and use this area just to attach the bacteria. And however, if the sample is from the patient, such as the fluid of the patient or the wound of the patient, get from the surface of the patient, it is the same. You need to use a clean stool to get some sample. Only a few, not too much, only a few of the sample. And do not use the cotton stick, because the cotton stick, there are some fibers of cotton, and the fibers of the cotton can attach to the slide in the saline, the water, the fluid. Then it will make you a little difficult to observe the bacteria. So please remember, do not use the cotton stick. Yeah. Actually, in the laboratory or in the hospital, they will provide you some tool like this. Yeah, now he get the sample.
And touch the edge of an isolated colony on the Petri plate. Place the loop into the drop.
Now you see, when he gets the sample and just rub the sample into the saline for a few times to make sure that the sample, the bacteria has been spread into the water or into the saline. Yeah.
On the slide and swirl it around to distribute the cells evenly, so the cells are one layer thick. Using cells from a single colony is important, as this should represent only one cell type rather than a mixture of cells. Ideally, the culture should be between 18 and 24 hours old, because
actually, he introduced, if you try to get bacteria from the cultured in the laboratory, it says it's better to get a single clone. But if you are in a hospital, you cannot get a single clone, because all the bacteria, the samples, are from the sample which collected from the patient, right? So, yeah.
Because older cells may show variable results. Alternatively, you can use a collection swab from a clinical specimen or environmental sample. Do not heat the slide. Wait for the sample to air dry. Bacterial cells.
Okay, and when we drop the sample into the saline, some students in the laboratory, when they do this experiment, they are so hurry. They put the slide on the fire to make it dry. So that is forbidden. It need to let it dry in the air by itself. It may take 10 minutes to half an hour. So just let it dry in the air.
Cells are then fixed to the slide using methanol. Fixing cells is important, as it prevents the cells from washing off during the staining process. Methanol fixation is recommended rather than heat fixation, as methanol preserves the cell's morphology. Heating the slide will cause cell distortion, could increase cell debris, and may cause erroneous gram reactions. Place or hold the slide over a paper towel, and flood the slide with absolute methanol for 2 minutes. Alternatively,
and they just told you to use some methanol to fix the slide. Okay.
You may dip the slide into a Copeland jar filled with methanol. Once 2 minutes have passed, tilt the slide and drain off the excess methanol and let the slide air dry. Do not wipe or blot the slide.
And when you try to make the slide dry, make sure there are two sides of the slide. You see a slide have front side and back side, and you must make sure your sample are on the front side, and do not wipe the slide by a tissue. You know the tissue paper, right? Do not wipe the slide by a tissue, otherwise it will remove your sample. So just let it dry by itself in the air.
As this can remove cells. Do not heat the slide. Cover the slide with a primary stain, crystal violet, for 1 minute. Crystal violet is a dark blue to purple dye.
So the first reagent, is the crystal violet. The color is like it's the dark blue. Just cover it and wait for
Stains all
2
cells.
minutes.
Rinse the slide with deionized or tap water to remove excess crystal violet.
And then you can use some water just to slightly wash the slide. Do not wash it very quickly. Otherwise, if you wash it with a very heavy water, with lots of water, it can make your samples flow away with the water. So be careful when you try to wash the first reagent.
Flood the slide with grams iodine for 1 minute. The negatively charged iodine molecules act as a mordant and link to the positively charged crystal violet. This traps the crystal violet in the peptidoglycan layer. Once 1 minute is up, rinse off the grams iodine with deionized or tap water.
Then wash it again.
Tilt the slide at an angle over the sink or tray and add the decolorizer dropwise until the violet color stops running. When the solution runs
So the third reagent just to remove all the first two, the color of the first two reagents. Until you see the solution runs clear. You see this is clear.
Immediately rinse the slide with deionized or tap water to neutralize the decolorizer. The decolorizer is composed of a ratio of alcohol to acetone. The ratio is normally a personal preference, with faster decolorizers containing higher concentrations of acetone and slower ones containing higher concentrations of alcohol. When decolorizer is added to the smear, lipids are extracted from the cell wall of gram-negative bacteria. Lipid extraction causes an increase in cell wall permeability, and the cell becomes leaky, resulting in the loss of the purple dye mordant complex from the thin peptidoglycan layer. In contrast, the effect of decolorizer on gram-positive bacteria is dehydration. This decreases the cell wall permeability, causing gram-positive cells to retain the crystal violet grams iodine complex. Note that a thick smear will require more decolorization.
So as the gram-positive bacteria has a thick glycogen, so the color of the reagent can stay in their cell wall. Do you remember the first reagent? It is dark. It is blue. Let me see. Where is the first one? This one. You see, the small picture. It shows this is the first reagent. It is dark blue. It is purple, deep purple, and deep blue color, right? So this one, it can be kept in the cell wall of the gram-positive bacteria. It cannot be removed away because the structure of the cell wall.
Than a thin smear. Be sure to stop the decolorization process immediately after the blue dye stops running off the slide. Beware. Over-decolorization can result in false gram-negative results.
So it means this, let me show. colorization. This step is to remove the extra color. So it means do not wait for a long time. Do not do the step for a long time. Otherwise, maybe all the color, not all, but some of the color can be faded, can get fade. Get fade means the color will be light and more and more light. Then maybe you cannot distinguish the true gram-positive and the gram-negative bacteria. So where do we? Okay, this one.
Gram-positive results. In essence, gram-positive cells will be blue-purple, and gram-negative cells will be colorless after decolorization. The final step utilizes a counterstain of a different color to differentiate gram-negative from gram-positive cells. Cover the slide with safranin or carbofuscin for 1 minute. The gram-negative bacteria will absorb this dye, causing them to stain pink to red. There will be no effect to the gram-positive cells since they retain the initial crystal violet stain. Rinse the slide with deionized or tap water. Do not wash the slide excessively. Allow the slide to air dry by tilting it onto a paper towel or over a sink and allowing it to air dry. Alternatively, gently dry the slide by blotting it using a lint-free bibulous paper. Do not use a wiping motion, for this can remove the smear. To summarize, crystal violet exposure, 30 to 60 seconds. Iodine exposure, 30 to 60 seconds. Acetone alcohol decolorizer, only enough time to rinse away the blue stain. Safranin exposure, 30 to 60 seconds. The slide can now be viewed under the microscope.
So yeah, in the summary now, just now you can understand the first reagent, the violet. This reagent it stain the cell wall of the gram-positive bacteria. Yeah, this is most important reagent for the gram-positive bacteria to give it, to make them the color of the blue or the purple. Yeah, and this one, the last one, it gives the color of the gram-negative bacteria the color of pink or red. Yeah.
The slide can now be viewed under the microscope. First, focus on the image using the high dry objective lens marked 40x. Then, without removing the slide, switch to the high power oil immersion objective lens that is marked 100x. This will result in an overall magnification of 1000x. Here you see two organisms routinely used for quality inspection at Hardy Diagnostics. The purple gram-positive cocci are Staph aureus. The pink gram-negative rods are E. coli. Hardy Diagnostics offers an improved formulation of gram stain called Gram Stain Advanced. This kit produces superior stain results.
Actually, nowadays the reagent for stain the bacteria for the gram stain, you can buy it from a company. Yeah, or there are so many companies they can provide the reagent, or we call it a kit. Kit means a box which contain all the reagent you need. Just you see the violet and the gram sliden and the alcohol or the methanol or something like this. So yeah, you just get it and follow the protocol, and then you can get the result.
With bright, vivid colors, even for bacteria that are difficult to stain. To test the quality of your stain and staining technique, Hardy Diagnostics offers quality control slides, the Q-Slide Gram, that are pre-inoculated and methanol fixed with gram-negative and positive bacteria. Thank you for watching this gram stain demonstration. Hardy Diagnostics is your complete one-stop.
Okay, so that is the procedure to do the gram stain. And here I have another link, another video which also show how to do the gram staining, but actually it's almost the same, just like just now you see 4 kinds of reagents. Yeah, put them one by one and wash them. Yeah, just wash them slightly, and finally observe the result under the microscope. And this experiment is better to do it in the laboratory because the teacher will capture some kind of the as we showed here just now. Yeah, we show some common gram-negative bacteria and some gram-positive bacteria. The teacher will capture the bacteria in a plate, and then they will distribute the bacteria to each student, and you will perform the experiment totally by yourself, and finally you will see the result. However, by the online class, we can just watch the video just to show you how to perform the experiment. So yeah, that it is actually in the PPT, yeah, in the slides, you see here, this one, yeah, I think this one is totally wrong. We cannot hit it on the file, right? Just we read it, we watch it on the slide and in the video. So in the slides, yeah, it also shows some picture to let you know how to perform the gram staining, but just like the video has been showed it. Yeah. So finally observe it under the microscope and you will get the result. So this is the most simple test or diagnose for the distinguish the bacterial, the gram staining. Yeah, that is so. For the second experiments, for the second laboratory, you don't need to write lab report for this part. Yeah, we just need to write down your lab report for gram staining. Yeah, all of this, the objective, materials, principles, and then step, and the mechanism. Mechanism means how to, why it can make the color for gram-negative cell wall and gram-positive cell wall, and then write down the steps, write down the procedures. Okay, write down the procedures. Yeah, you need to finish it like this. So please submit your lab report on next Monday. Yeah, on next Monday. Remember it. So this is today's class. Yeah, class is over. And what's more, I want to say, yeah, in the end of the class, I want to tell you that as you know, we have the class schedule, and as sometimes we have theory class, sometimes we also have the lab class, and as my busy schedule, I need to do research in the laboratory. So I want to say that after this lab class, tomorrow morning, I will send the video of the theory class again in our Dean Talk group. Please watch the video of the theory classes. My tomorrow morning to me it means this evening, 7 o'clock in your time, Beijing time, 7 o'clock in the evening, right? Okay, so yeah, then I hope you understand it. So I think class is over today. If you have any questions, then just leave me message in our Dean Talk group. And yeah, I will send this website and also these two links, yeah, of the procedures in our this one slides in our PPT. Sorry. Yeah. Okay, it works. Yes. And you can watch the video after the class. Okay, see you. Bye.
OK，老师，See you，老师，Thank you，
谢谢老师，Thank you，
Thank you everyone，谢谢老师，拜拜，Thank you，老师，
拜拜。
拜拜。
再见，老师。